Hey guys, welcome to our third lecture on the nervous system basics. Last time we met, we had talked about the fact that a neuron at rest, one that is not doing any signaling, has a membrane potential, and that membrane potential is basically a way of saying it has a voltage, that has this electrical state where the inside of the cell has a difference from the outside of negative 70 millivolts. And we're going to utilize this difference to send electrical signals. Excuse me. But first of all, we got to figure out how this electrical difference is established. So, how do we establish a membrane potential? All right. What I'm looking at here is a neuron. All right, where it says inside cell, that cell is a neuron. And you can tell that this is the neuron's plasma membrane right here. Check out all those beautiful phospholipids with their fatty acid tails. Very nice. And we have an integral protein right here. You have met this integral protein before. What is it called? Does anybody remember? Yes, it is the sodium potassium pump. We talked about it a long time ago when we were doing the cells and transport lecture, and that sodium potassium pump was an example of primary active transport. And what this guy does is it is going to smash a molecule of ATP, converting that ATP into ADP. And when it does so, the energy that's released is going to move 1, 2, 3 sodium ions out of the cell, and then it's going to move 2 potassium ions into the cell. So 3 sodiums going out, 2 potassiums going in. Now, what I want you to notice here is that sodium and potassium ions, they are cations. Think back to chemistry class. Remember that there are cations and there are anions. The T in cations looks like a plus, sort of. So they're cations, they have positive charges. So every time the sodium potassium pump does its job, breaks down an ATP, it moves 3 pluses out and only 2 pluses in. This is a fundamental imbalance here. This makes the cell more negative on the interior. The interior is getting more negative. And it is bringing the interior of the cell close to that value of negative 70 millivolts. Okay, the sodium potassium pump is one of the players in the generation of our resting membrane potential. But there's more. There's always more, right? There's also another type of integral protein we find in our neuron cell membrane. Another type of protein. So I'm inside the neuron here, where it says cell interior, that's inside the neuron. In the membrane, here's our plasma membrane again. In the membrane, we have another type of integral protein called a potassium leak channel. We've actually met these guys before as well when we were dealing with facilitated diffusion. And what these guys do is they allow potassium to leak out. Potassium is higher in concentration inside the cell, so potassium tends to go out. As potassium leaks out, well, it's a cation, right? It's a positive charge leaving that causes the interior of the cell to become more negative. So right now we have 2 proteins that contribute to this resting membrane potential. The action of the sodium potassium pump and then the presence of these potassium leak channels is going to cause the interior of the cell to be more negative than the exterior. Okay, good. In fact, if it's up to these 2 proteins, the interior of the cell will have a resting membrane potential of negative 90 millivolts. Wait a minute. Is that what we said before? No, we said the resting membrane potential. We said that resting membrane potential was equal to negative 70 millivolts. But these two proteins give us a membrane potential of negative 90. Okay, there must be something else going on. Let's figure out what it is. So there are actually also another type of integral protein in the membrane of our neuron called the sodium leakage channel. That's a sodium leak channel right there. Now check out what sodium is doing. Sodium is going in. It's diffusing inward. And the diffusion of sodium inward brings a little more positive charge inside, which brings our resting membrane potential to that value of negative 70. Bottom line, there are three things that impact resting membrane potential: sodium-potassium pump, potassium leakage channels, and the sodium leakage channels. These guys are ubiquitous within the neuron. They're found in the dendrites, the soma, and the axons. So they're all over the place. So the whole neuron is going to have a resting membrane potential. Now you might be wondering, okay, what's the point of me learning this? Well, as we're going to see, changing the resting membrane potential is how we send electrical signals. All right. We're doing well here. We're doing well. Okay. Same picture we had earlier. Inside the axon here, it's more negative than the outside. How much more negative? 70 millivolts more negative. So we say the inside has a voltage of negative 70 millivolts. All right, there's a word for this. There's a word for this electrical difference. A neuron is said to be polarized. The word polarized means there is a difference. I think it would be fair to say that we have a very polarized political system in the U.S. right now. There is a big difference between the sides. And we use this term to describe a neuron. A neuron at rest is polarized. There is an electrical difference between the inside and the outside. All right, keep that in mind. Tuck that in your brain for a second. We're going to come back to that. All right, reiterating something we've said before, we're going to use electrical signals to go from one end of a neuron all the way down to the other. So electrical signals through the dendrites and through the soma, and then through the axon. Now notice that I chose different color arrows to kind of represent my electrical signals. Why is this? Well, if you remember from before, in the dendrites and the soma, we're going to have a type of electrical signal called the graded potential. Whereas in the axon, we're going to have a type of electrical signal called the action potential. Now, both of these electrical signals are waves of change in the membrane potential. Waves of change in the membrane potential, okay? That's what these guys are. That's what these guys are. All right, I'm doing my third video of the day right here. So I feel like I am going to be misspeaking like crazy. I don't mind though. Bear with me, please. All right. Two types of electrical signal that we're going to deal with in our neuron. One in the dendrites and the soma, that's called a graded potential. One in the axon, that is called an action potential. Okay, Good. So we have a graph here. We have a graph. Check out the axes of the graph. The y-axis is membrane potential. The y-axis is membrane potential. Okay? The x-axis is time. So here's membrane potential on the y-axis. Then time is our x-axis. And now we're starting out here at rest. So like for this period right here, the cell, the neuron is at rest. Unchanging membrane potential of negative 70 millivolts. Okay? Now, what we're going to see here is there's three basic ways the membrane potential can change. So we're starting at rest. And remember, if we are at rest, one of the things we said was that the neuron was polarized, meaning there is a difference between the interior and the exterior. All right, the first change we see here starts right here. First change we're going to see starts right here as the membrane potential swoops upward and becomes more positive. This is known as depolarization. All right, let's break down that word for a second. Depolarization. I cannot spell today. Why can I not spell today? Depolarization. Should we have spelling on the next exam? It's a good idea. All right, what does the prefix D mean? If you are dehydrated, of course you are without water. You have reduced water. So I'm going to write the word reduced down. There we go. Polarization, which we already know, meant there was a difference. So if we're depolarizing, we are reducing the difference. Notice that in this graph, we're going up. We're becoming more positive. Remember, the outside of the neuron was positive. The inside was negative. So if the inside becomes more positive, it gets more like the outside, which means there is less of a difference. And so this is called a depolarization. Now, our graph went up, it leveled off a bit, now it's coming back down. This return is called repolarization. Right? We are having something again. If we're establishing a difference again, that is called repolarization. And we're getting back to the resting membrane potential, that RMP, right there. Okay. All right. Another change happens right here. In this case, our membrane potential gets more negative. It's even more different. That's not very good English, but it's the way I like to say it. More different than the outside. So because it's more different, we are saying it is hyperpolarization. So these are the basic ways in which we can change the membrane potential. We can depolarize it, and if we depolarize it, we can repolarize it, and then we can also hyperpolarize it. Notice we don't have a term for the return to normal from hyperpolarize. That's okay. We'll survive without that. All right, so these are ways I can change my membrane potential. And if I go back for a second, remember the electrical signals in the dendrites and soma and in the axon are waves of change. So they're either waves of depolarization, waves of hyperpolarization. We're going to see. Let's keep going. All right. What I have here at the top, we're going to start at the top here, guys. At the top, I have a synapse. This is a synapse up here. I have an axon terminal right there. Axon terminal is filled with synaptic vesicles, which contain neurotransmitters in them. this axon terminal is right up against a dendrite on a postsynaptic neuron. Now what's going to happen is an action potential, the word impulse here is referring to the action potential. The action potential goes down this axon and prompts the release of these neurotransmitters, their exocytosis. The neurotransmitters then diffuse across the synaptic cleft and bind to these receptors. And these receptors are ion channels. And they are said to be transmitter gated. That means they'll be open or closed depending on whether or not a neurotransmitter is bound to them. So when the neurotransmitter binds to these guys, they open up. And look what sodium does. Sodium rushes in. Sodium is a cation. When sodium rushes in, my interior of my neuron here, well, it is going to get more positive. And we see that in my graph here. It goes up. It gets more positive. The word for this positive change is depolarization. So the point I'm trying to make here is to show you that when one neuron releases neurotransmitters onto another, the receiving neuron can depolarize in response. It can have this change in membrane potential. This spot on the neuron right here depolarized. And we graphed it right here. This depolarization has been labeled as an EPSP. EPSP stands for excitatory. We'll see why shortly, okay? Postsynaptic, because what neuron is experiencing this depolarization? The one getting the neurotransmitter, the postsynaptic neuron, and then the last P is potential. Because what are we changing? We're changing our membrane potential. Okay, one of the things we can do is use neurotransmitters to depolarize a neuron, and that depolarization is called an excitatory postsynaptic potential. Okay, good. Let's see another example. I got another axon here, but now this axon is releasing different neurotransmitters. Different neurotransmitters in here, these purple guys here. They're binding to different receptors on my postsynaptic neuron. Again, these receptors, they're integral proteins, they're transmitter-gated ion channels. And when these guys open up, well, look who's going through. It's chloride. And in this case, our postsynaptic neuron, our recipient, the one binding the neurotransmitters, it is now getting an influx of chloride. Chloride, of course, is an anion. It is negatively charged. So the interior of our cell down here is going to get more negative. I see that in my graph as I go from my resting value of negative 70 down towards negative 90. And that is an example of hyperpolarization. So we now see that one neuron releasing neurotransmitters onto another could also result in the hyperpolarization of the postsynaptic neuron. Okay, good. All right, here's where it starts to get interesting. Let's suppose that right where I've drawn this green lightning bolt, that is where neurotransmitters are going to bind to the dendrite. That is where neurotransmitters are going to bind, and they are going to create a depolarization there, an EPSP. Now, here's something to realize. When we get the depolarization here, it will spread. The depolarization will spread. But there's a question mark involved here. As it spreads towards the axon hillock, what will happen to it when it gets there? Will it still be big? That's the question. If we create a depolarization here and it starts to migrate, it will get smaller as it migrates. If it gets to the axon hillock, and it achieves this value called threshold is a value of negative 55 millivolts, then the action potential will send its own electrical signal. So the action potential, the axon has this special membrane potential that if we get to it, it sends an electrical signal. And that special value is called the threshold potential. Its value is negative 55 millivolts. It only matters for my axon. So, if we stimulate a dendrite and we generate an EPSP and it travels and travels and travels and travels, as it gets to the axon, if it's big enough to get to this threshold potential, and by the way, this little guy right here was not big enough. If it's big enough to get it to negative 55, then our axon is going to send its own signal, which is an action potential. All right, don't worry. We still have a lot of unpacking and figuring out to do. Okay? A lot of unpacking, a lot of figuring out to do. All right? Let's backtrack for a second here. If a channel in a neuron opens and sodium rushes in, that will make the membrane potential get more positive. The word for that change, it is a depolarization. A depolarization is an example of an EPSP, an excitatory postsynaptic potential. Potential is used here because we're dealing with a change in the membrane potential. Postsynaptic because we're talking about the postsynaptic neuron, the one that gets the neurotransmitter. Excitatory because if the membrane potential gets more positive, that is going to bring us towards threshold. That's why we use the word excitatory here. All right. Now, the other type, and I don't think I talked about this earlier, it was on the slide, but I forgot to mention it. When the membrane potential gets more negative, so for example, if there's influx of chloride, we are doing some hyperpolarization. Remember that word? Because we're getting more different than the outside. We're doing some hyperpolarization. If we're doing hyperpolarization, we're getting farther away from that magic number called threshold. And because we're getting farther away from that number where the axons do something, this hyperpolarization is known as an IPSP, an inhibitory postsynaptic potential. Okay, we're figuring out how neurons send signals, okay? Just going to draw a neuron real quick. Give me a nucleus, give me some cell body, give me some dendrites. Quickly drawing a neuron. Bear with me here. So I've drawn nothing but dendrites so far. And now I'm going to give this guy an axon. Ooh, got kind of skinny there. There we go. There's my neuron. Let's suppose there was another neuron. And here's his axon. There it is. I don't know why I made him so curvy right there. There we go. Doesn't matter. Actually, you know what? I don't like that one. I'm going to redo him. Let's redo that one. There we go. That's better. So, this guy right here is my presynaptic neuron. And my yellow guy is my postsynaptic neuron, okay? My green guy is going to release neurotransmitters. Ba-bum, 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 ba-bum. They bind to this dendrite here. They cause sodium channels to open. Let's do sodium. Sodium then is going to rush in. Sodium's going to rush in whoosh, right here. Okay, originally my neuron, my resting neuron here, the yellow one, had a membrane potential of -70. When the sodium rushed in, this spot right here depolarized, got more positive. And then that depolarization is going to spread towards the axon hillock. As it spreads, it gets smaller. That's what happens to waves when they move, right? As waves travel, they get smaller and smaller. If my graded potential, that's what this is here, if this wave of depolarization is able to change the membrane potential right here to -55, then my axon is going to fire and send a signal, an action potential that goes down this way and causes the release of neurotransmitters down at the end. We still have to figure out more about this. This is like the bare bones of how it works. But we're going to unpack it even more. All right. How many times can I say the word unpack in one lecture? How many times can I say the word good? I keep saying that. Good, good, good. I don't know why. All right. Let's talk about graded potentials. Actually, you know what? I'm going to end this lecture right here. I'm going to do 4 lectures for nervous system basics. I'm going to stop this one right here. We'll do another one later. Watch this one twice maybe before you go on to do the next one. All right? Okay, cool. See you guys next time.